In this video, we're going to talk about the concept of homeostasis. And to illustrate it, we're going to actually use some of the metabolic systems we're going to be unpacking in future videos. So it's sort of a twofold purpose. But homeostasis, it turns out, is one of the coolest things that our cells do. It's a really important framework to have in mind as we're trying to understand metabolism. So let's tackle that. So in terms of just a definition, homeostasis is the ability of an organism to keep a stable internal environment despite external changes. And the fact that we can do this is actually something that basically means we're not at equilibrium. We're not a boring system that's dead. Instead, we're a living system. And we're going to look at two really cool ways that we see homeostasis at play. The first is in terms of the internal energy state of, say, a cell. And then we're going to talk at a physiological level about blood glucose concentrations. How do we maintain those at a fixed level? And then in future videos, what I'd like to do is we're going to talk about sort of the what here. Oh, look, we maintain an internal energy state that's fixed. And then in future videos, we'll ask, well, how is that done in terms of detailed molecular mechanisms? All right, so let's talk about internal energy states. So we've talked about ATP being this important this energy currency. And let's imagine for a second that you say, go out on a run, or you stand up quickly, or you start doing something where you're going to continue to sustain that activity. So what happens in your muscle cells is your ATP concentration starts to go down because you are converting ATP into ADP to cause your muscle fibers to contract, let's say. Now, immediately before any other metabolic process happens, you start seeing this reaction happen. This is catalyzed by the enzyme adenylate kinase, which is just kind of floating around in the cell all the time. What happens is the concentration of ADP starts to go up. And what adenylate kinase does is it catalyzes the reaction of ADPs combining, basically taking a phosphate off of one, sticking it onto the other, forming ATP and AMP. And so this is just Le Chatelier's principle. I've said in previous videos that Le Chatelier's principle is the answer to most biochemistry questions. Here's an example. The concentration of ADP goes up, goes up, goes up. What that does is it drives this reaction this way. And look at that. Voila, you're generating more ATP. So, okay, you're doing this, that's great, you're recovering your ATP, but you keep doing activities. And eventually, if you did this, right, you'd have no ATP left, right? This is not replacing the ATP, it's just recycling some of the products. It's like betting, it's taking the output and using some of it to make more ATP. So you need to make some more. How do we do that? Oops. Well, it turns out we turn on this process here, which is glycolysis. What does glycolysis do? It takes glucose molecules, it cuts them in half, uses the energy from cutting that in half to generate 2 ATPs. And so you're getting more ATPs. But here's a question. How does a cell know that it needs to run glycolysis? There's a variety of mechanisms, but one of the most important is actually this, that AMP activates glycolysis. So we are now making more ATP because our AMP concentration starts to go up. Let's say you keep doing things. You're not going to stop. You're going to keep going further. You're going to keep doing whatever activity. If you're on a run, you're running more than 100 meters or so. Well, now you start to kick off a whole nother level of processes. And so what do you get here? This is the citric acid cycle plus oxidative phosphorylation, but basically you start taking pyruvates and you start chopping them and generating carbon dioxide. So you're fully oxidizing pyruvate, and in the process releasing a bunch of ATPs. But how does the cell know that it needs to do that? Well, it turns out that in some part at least, this is activated by pyruvate. So as pyruvate concentration goes up, it says, hey, start running this process here. What we're seeing here are a whole bunch of homeostatic mechanisms. Homeostatic mechanisms. So homeostatic mechanisms. What do I mean by this? Well, let's look at this. So when your ATP concentrate, or ADP, you start consuming ADP and producing ATP, you have a restorative mechanism here that basically says, hey, ADP concentration shouldn't be going up. If ADP concentrations are going up, do something with that. Make your ADP concentrations start going down, right? This is homeostatic. Keep ADP concentrations at a certain level. When we get to AMP, what we're saying here is recall that this is activated by AMP. So this is saying, man, AMP concentrations are going up. Start doing a mechanism that's actually going to drive AMP concentrations down. At the end of this reaction, we're starting to build up pyruvate. This is a reaction that says, let's start consuming that pyruvate. Wow, I just totally missed with my arrow. This is saying, start consuming pyruvate. Pyruvate concentration should be low. They're building up, turn on a mechanism that gets them lower. And then concentrate, this keeps happening, right? Glucose concentrations, we actually have mechanisms to keep our glucose concentrations high. As those start getting depleted, we're going to start tapping into sugar reserves. And it goes on and on and on. Across metabolism, what we see are homeostatic mechanisms. As the concentration of a molecule starts to get out of whack, restorative things pop in to try to push it back into the correct concentration. And the mental picture I think that's really helpful for this is that of biological cruise control. So if you're driving a car and you set cruise control, what that means is the speed of your car is going to stay the same, whether or not you're going uphill or downhill, or you've got wind blowing on you. And that's because of a feedback loop of some sort, right? You've got a computer that's sitting there measuring the speed of the car. That's coupled into your throttle so that as your speed starts to slow down, the throttle starts to increase. As your speed starts to speed up, your throttle decreases, right? And we see that all throughout biochemistry, where there are things like keep ATP steady. That is a really high level thing that cells try to do. And have all these mechanisms in place that are just aimed at keeping ATP concentration steady. And the way that this is achieved is through the sequential activation of specific pathways. And when we dig into biochemistry, we're going to see this over and over and over in like glorious, gritty detail. But at a high level, this is what they're doing. They're saying, man, ATP concentration is going down. Let's make some more. What are we going to do? Well, we're going to keep making more. We're going to turn on more and more complicated stuff to keep that concentration of ATP high. Okay, so that was at the level of, say, an individual cell. What happens at the level of, say, our whole physiology, our blood glucose levels? And the idea here is we can start saying, oh, there are some parallel sorts of ideas at play across these physiological scales, from individual cells to whole physiology. So, again, like many things in these videos, we'll unpack these in more details later. But for the moment, I'm going to draw a very schematized version of our entire fuel system. It consists of muscle cells, myocytes, islet cells from the pancreas, hepatocytes in the liver, adipocytes, or fat cells. These are all connected to one another via the bloodstream, so things circulate in the bloodstream. One of the things that we work really hard at is keeping our blood glucose concentrations in a narrow range. And this is shown here on the left in an acute sense. So if you eat meals, your blood glucose goes up. If you exercise, your blood glucose might go down. But if you measure blood glucose over your day, even with these spikes, what you'll see is the concentration fluctuates in a relatively narrow range of, say, 4 to, say, 6.5 millimolar. It's just going to fluctuate between that level. And it also is maintained in more of a sort of chronic sense. So if you're not eating, for example, this is showing blood glucose concentrations as a function of days of starvation. And what you can maybe appreciate from this is you start out at a relatively high level, it drops, and then it's going to remain the same even if you consume no food. And so the idea here is our body is working hard to keep this blood glucose concentration inside of a narrow range. If it goes outside of this range, and particularly if blood glucose concentration is high and remains high, you have all kinds of the sorts of disease states that are associated with diabetes and metabolic disorders. That's basically because cells are tuned to expect a particular concentration of glucose, and as that concentration gets out of whack, cells actually turn on incorrect regulatory pathways, and that leads to the poor health the poor health outcomes in a situation like diabetes. Okay, so what we want to ask here is not sort of the consequences of falling out of it, but just how is this homeostasis achieved? And what we're going to see here is a set of feedback loops. Those feedback loops involve basically interaction between a couple of classes of molecules. We're going to have glucose. So this is our sugar, this six-membered ring sugar. It can form arrays with a molecule called glycogen. And we're going to do an entire video just on glycogen. But for now, all we need to know is that, oh, look, it is an array of a bunch of different glucose subunits that are glued together. This allows cells to store glucose for later, right? Just glue it together and we can pack it away for later. The other major player that we're going to see are these things called fatty acids. So this is an exemplar of a fatty acid, long acyl chain, carboxylate side on it. These fatty acids can be glued into triglycerides, which is their storage form. And this is basically three different fatty acids glued together, which I'm going to draw schematically like this, actually to make them more compact. But it's basically 1, 2, 3 acyl chains glued together. These can actually also be made into ketone bodies, which is an approximation for glucose. In many cell types, ketone bodies can substitute for glucose in an emergency. Okay, so what I'm going to do is talk about this homeostatic mechanism at the level of these cell types. Not in any detail. I'm just going to say this is what happens. And then again, in future videos, we'll start asking in more detail, like how is it that this actually happens? But for now, we are going to try to keep our blood glucose high, and we're going to ask, how is it achieved by these organ systems? Okay, so let's imagine that we just have muscles that are consuming energy. So they're taking glucose and they're burning it to do stuff. One of the first places that they can draw from for this is from their own internal glycogen stores. Oh, actually, I meant to say that. The different cell types have different components in them. So muscle cells, myocytes, they have glycogen in them. Liver cells have glycogen and a small number of triglycerides, and then adipocytes have a bunch of triglycerides. Okay, so we have a muscle cell. It's consuming glucose. That glucose can initially come from its internal glycogen stores. It can also draw glucose directly from the bloodstream. And once it starts doing that, your blood glucose concentration is going to start to go down, right? Because the cells are consuming it, they're turning it into carbon dioxide. That glucose is going away forever. So the pancreas, islet cells in the pancreas can detect changes in blood glucose concentration. What they do when this happens is they start dumping out a hormone known as glucagon. The hormone is simply a molecule that signals across the body. So it's produced in one cell type and it's read out in other cell types. And in this case, hepatocytes can actually read, they can read, glucagon's around. They can sense the presence of glucagon. And so they start taking their glycogen, breaking it down and dumping it into the bloodstream. And so what we're seeing is a homeostatic mechanism here, right? The hepatocytes are making more glucose to basically replace the glucose that's being consumed by the cells, by the muscle cells, keeping blood glucose concentrations about the same. Okay, now let's imagine that you continue to consume energy, or it's a little bit subtle here. It also depends on the rate of consumption. So if you're consuming really quickly, your body will tend to prefer glucose. If you're consuming at a moderate rate, muscles will very happily burn fatty acids. And so let's imagine now that we're consuming both glucose and fatty acids. Well, when your glucagon is elevated and you're consuming fatty acids, your adipocytes will start mobilizing their stores, right? So your triglycerides will start being broken down, dumped out as fatty acids. They can then go into your muscle cells. And so we're seeing, again, a homeostatic mechanism. As fatty acids are being consumed, fatty acids are coming in from outside. Okay, cool. Let's imagine now we enter starvation conditions, or we've gone for a very, very long time working really hard, and our glycogen stores are gone. So this could be starvation. This could actually be even, if you've heard of athletes that hit the wall, part of that correlation with a distance athlete hitting the wall is their glycogen stores get really, really low. Well, what's a cell to do? Turns out, I should not have put an X over there, our glycogen's gone. One of the things that this triggers is through this sustained exposure to glucagon, right? Because blood sugar is continuing to be low, we start seeing a process called ketone body formation happening over here, where instead of liver cells, they'll start importing fatty acids, converting those fatty acids into these ketone bodies, these small sugars, which then can be used by muscle cells. So again, this process is a non-homeostatic thing, right? This is sucking energy out of the cell. This is burning it, turning it into carbon dioxide, going away forever. But we have a bunch of restorative mechanisms that are like, no, we need to keep our blood sugar high. We need to keep fatty acids around, and those are mediated by, say, the liver, in this case, creating more ketone bodies. Okay, last thing I want to point out, and then we'll be done with this. And then in the next video, we'll start digging into a little bit more detail on this, actually. But now imagine that you've eaten a bunch of sugar. We've gone out of our starvation conditions. We've got some glycogen around, so we're back to our starting state. But now we've eaten some sugar, so food has come in. Glucose is coming in. That can be sensed by the pancreas as well. And what the pancreas does in this case is it dumps out the hormone insulin, which says, hey, blood sugar is going up. We should probably deal with that. How do we deal with that? Well, what we see is hepatocytes start importing glucose and sticking it into glycogen. We start seeing adipocytes importing glucose and turning it into fatty acids and ultimately into triglycerides. And we see muscle cells, if their glycogen is depleted, start taking up glucose and sticking it into glycogen, right? Blood glucose is going up. Homeostatic mechanism, push it back down. How do we push it back down? We start storing it up for later. And if blood glucose continues to be high and blood insulin continues to be high, we start seeing processes like this one where glucose gets imported and then diverted into fatty acids. And actually you get fatty acid synthesis. And then fatty acid synthesis can be made into triglycerides. Triglycerides get exported. What happens with those triglycerides? Well, now they get dumped into adipocytes. It's a little bit of a complicated mechanism, but overall, we're again seeing a homeostatic mechanism, right? Blood glucose is high. Blood glucose remains high. What do we do as we continue to deal with that glucose? We start storing it as glycogen and then ultimately storing it as fat. And so hopefully what we've taken away from this is that much of metabolism is homeostatic. All these complicated mechanisms we're going to talk about and see can be understood, or most of them can be really understood as trying to restore the system back to its initial state, to try to restore ATP concentrations or say blood glucose to a fixed level. So biological cruise control. What we see is within cells, pathways activate sequentially to keep the concentrations of metabolites steady. ATP is one of the sort of mega things that's trying to keep steady, but it's mediated along the way. As pyruvate concentrations come up, start breaking down pyruvate. As AMP concentrations start going up, start figuring out ways to turn AMP back into ATP. And then at the level of blood glucose, this is achieved by homeostatic mechanisms that are spread across multiple organs, coordinated by hormones. But again, you have this blood glucose gets out of whack, turn on systems that bring it back into case, bring it back to its starting level. Okay, so that was just a really quick overview of two major sets of metabolic systems inside of cells and across organ systems, but viewed through the lens of homeostasis, and we're going to see that apply again and again throughout biochemistry. Really look forward to digging into some specific pathways with you very soon. We're laying a lot of important groundwork for that, but until then, I hope you continue to enjoy thinking about biochemistry.